Let's continue with lipids. Now we are going to learn about the conjugated lipids, compound lipids or heterolipids. Compound lipids or heterolipids are the lipids which remain linked with non-lipid molecules such as proteins, oligosaccharides and phosphates. That is something which we have already learned. So glycolipids, we have already learned about them. These are the sugar lipid complexes in the one or more monosaccharide residues are glycosidically linked to a lipid part. Sugar forms the polar head of the molecule. The lipid part contains fatty acid and spingenin. Glycolipids are primarily the constituents of biomembrane and nerve tissue. They are particularly abundant in chloroplast membranes and the myelin sheaths of nerve fibers. The commonest glycolipids are cerebrocytes and gangliosides. Now, let's learn about the cerebrocytes. Cerebrocytes are the simplest type of glycolipids. As the name implies, they are the glycolipids most abundant in the brain tissue, especially in white matter and myelin sheath. They contain a ceramide, spingenin attached to fatty acid, and one or more sugars. In most of them, galactose is the sugar, hence they are sometimes called galactolipids. In some cases, glucose is the sugar, and hence they are called glucocerocytes. Glucocerebrocytes. Large amount of cerebrocytes are found to accumulate in liver and spleen in Gaucher's disease, a congenital disease characterized by defective glycolipid metabolism due to autosomal recessive gene mutation. In this case, glucose cerebrocytes are most prominent in spleen. Several kinds of cerebrocytes have been isolated from brain and nerves. They differ from each other in their fatty acid content. Some examples are given below: cerebrine, cerebronic acid, kerosene, lingoceric acid, nervonic acid, and oxynervonic acid. Gangliosides are there. So gangliosides are the complex type of glycolipids commonly formed of galactose, glucose, sialic acid, fatty acid, hexosamine, and spingenin. They have been isolated from nerve cells, nerve ganglia, spleen, and stroma of RBC. Cells of cerebral cortex are rich in gangliosides. Large amount of them are found to accumulate in the brain of the patients of Tay-Sachs disease, a lethal hereditary disease associated with defective metabolic lipid metabolism due to autosomal recessive gene mutation. Gangliosides. Sulfolipids. These are the sulfurs containing compound lipids found in brain, liver, kidney, salivary gland, and tumors. They are most abundant in the white matter of brain. They are supposed to be the phosphoric acid esters of cerebrocytes. Sulfolipids have acidic properties and are capable of binding cations, so they can participate in the transport of cations across nerve membranes. Thus, they are necessary for the normal electrical activity of the nerve system. Derived lipids. These are the substances derived by the hydrolysis of simple and compound lipids. The most important one among them are the prostaglandins and isoprenoids. First one is the prostaglandins. Prostaglandins are a family of 20 carbon lipid soluble long chain organic carboxylic acids with a 5 carbon cyclopentane ring. Prostaglandins are a family of 20 carbon lipid soluble long chain organic carboxylic acids with a 5 carbon cyclopentane ring. These are the derivatives of a 20 carbon essential fatty acid, probably prostanoic acid, via arachidonic acid, which is unsaturated fatty acid found in liver and glandular organs. A prostaglandin molecule has a cyclopentane ring. Attached to this ring, there will be a 7 carbon chain and an 8 carbon chain. The former projects beyond the plane of the ring and the later projects above the plane. Prostaglandins were first extracted from human seminal plasma independently by Goldblatt in England and von Eller in Sweden. Prostaglandins are so named because a prostaglandin was first isolated from the secretion of the prostate gland. They are now known to exist in all mammalian tissues and biological fluids and are produced by many tissues. They are particularly abundant in the seminal fluid. Aspirin is a potent inhibitor of the enzyme prostaglandin synthetase. It mediates synthesis of prostaglandins from arachidonic acid. The analgesic role of aspirin is related to this property. There are two main classes of prostaglandins, namely E group and F group. Each of them has three subseries of or members: PGE, PG1, PGE2, PGE3, and PGEF1, PGF2, and PGF3. E group prostaglandins contain a keto oxygen group at C9 and hydroxyl group at C11 and C15. On the other hand, F group of prostaglandins have hydroxyl groups at C9, C11 and C15. Several types of prostaglandins are now artificially synthesized. Cystones are the major intermediates in this synthesis. The marine cylinderates gorgonians are the most prolific natural source of cystones. They live in large numbers in coral reefs. Cystones are the major intermediates in the synthesis. Prostaglandins have a broad spectrum of physiological effects. They are primarily regulators of hormone action. Apart from this, they affect nerve system, inhibit the production of gastric juice. Prostaglandins have a broad spectrum of physiological effects. They are primarily regulate hormone action. Apart from this, they affect nerve system, inhibit the production of gastric juice, stimulate smooth muscles, promote contraction and relaxation of uterine wall, induce labor during delivery, induce vasodilation or vasoconstriction, regulate blood flow and BP, modify the levels of cyclic AMP, antagonistically influence the action of some hormones, stimulate steroid production and insulin release, and promote the mobilization of Ca++ from bones. Prostaglandins are not to be released during the inflammatory response of damaged tissue. Natural and synthetic prostaglandins are sometimes administered to induce labor or abortion in human females and domestic animals. Thromboxanes, leukotrienes, and prostacyclins. These are the direct derivatives from prostaglandins. Thromboxanes, leukotrienes, and prostacyclins. Thromboxanes are produced in blood platelets, leukotrienes in leukocytes, and prostacyclins in the walls of blood vessels. Thromboxanes regulate the activities of blood platelets and promote vasoconstriction, platelet aggregation, and blood coagulation. The exact function of leukotrienes is definitely not. Prostacyclins are anti-thromboxane in action. They promote vasodilation and inhibit platelet aggregation. So isoprenoids are there. Isoprenoids are a vast group of biologically important lipids. They owe their name to their 5-carbon structural unit called isoprene. Isoprenes are the starting material in the biosynthesis of isoprenoids. Even though isoprenoids do not contain fatty acids, they are referred to as lipids simply because they contain isoprenes. The commonest isoprenoids include terpenes and steroids. CH2, C, CH, CH2, CH3. CH2 to C would be a double bond. So first one is the terpenes. Terpenes are unsaturated hydrocarbons with the formula C10H16. They are particularly common in higher plants and primarily responsible for the flavor and fragrance of essential oils and resins. They are also the essential constituent of gibberellins, plant growth factors, carotenoids, natural rubber, phytol component of chlorophyll, etc. Terpenes may be acyclic or cyclic compounds formed of repeating isoprene units. There are 4 major groups of terpenes, namely monoterpenes, sesquiterpenes, diterpenes, and tetraterpenes. The number of their isoprene units are 2, 4, 2, 3, 4, 6, and 8 respectively. The triterpenes, sequelins, and lanosterol are the precursors of cholesterol biosynthesis. Derivatives of terpenes are called terpenoids: camphor, menthol, terpenol, boronol, geraniol, etc. are examples of terpenoids. A group of oxygen-containing terpene are of special interest. They contain odorous isoprenoid alcohols which impart specific smell to plants. For example, farnesol, a terpenol, smells like lily. The monoterpenes geraniol, nerol, and corianderol smell like geranium or rose. These are found in large amounts in the attar of roses and are used for making perfumes. Menthol monoterpene is used in the confectionery industry. Camphor, a monoterpene ketone, is used as a drug. The terpenes phytol and renitol are of special importance. Phytol is an essential constituent of chlorophyll and vitamin K1. Phylloquinone. Retinol forms vitamin A. Next is the steroids. Steroids are a family of saturated and polycyclic hydrocarbons. Steroids are a family of saturated and polycyclic hydrocarbons. They have a cyclic ring or nucleus composed of 17 carbon atoms arranged in a system of 4 fused rings. Attached to the nucleus are 2 methyl groups in positions 18 and 19 and an aliphatic side ring in position 70. Steroids are cyclic derivatives of triterpenes. They contain neither fatty acid nor glycerols. Still, they are regarded as lipids on the ground that they are insoluble in water and soluble in organic solvents. Most steroids are OH containing tetracyclic alcohols, steroid alcohols, conventionally called as sterols. A sterol molecule has a central nucleus or body. It is formed of 4 fused rings, namely 3 cyclohexane rings and a terminal cyclopentane ring. Body bears a polar head group OH group at C3, 2 methyl groups at C18 and C19, a side chain at C17 and a double bond mostly between C5 and C6. Sterols of all species are synthesized from 5 carbon isoprene units. Sterols of animal origin are called zoosterols and those of plant origin are called phytosterols. Sterols are altogether absent in prokaryotes. Cholesterol is the animal sterol. Stigmasterol and cytosterol are plant sterols and ergosterol is the yeast sterol. Cholesterol is abundant in animal tissue, especially in liver, adrenal and gland and myelin sheath, either in the free state or as esters. Altogether with the phospholipids, it forms the molecular framework of cell membranes. It also serves as a precursor for the synthesis of bile acid, bile alcohol, sex hormones, adenocorticoids, vitamin D, etc. Ergosterol transforms to vitamin D to calciferol in the help of UV radiation. Biological functions of lipids. Lipids are biochemically very important because they are excellent source of reserve energy and essential structural molecules of cells. They are richer in energy potential and high in calorific value than carbohydrates. Lipids are biochemically very important because they are excellent sources of reserve energy and essential structural molecules of cells. They are richer in energy potential and higher in calorific value than carbohydrates. So, on oxidation, the energy yield from a given mass of lipid can be much higher than yield from the same mass of carbohydrates. Lipids are highly essential for the structural organization of cell membranes and some cell organelles and also for the normal functioning of cells. Some of their biological functions are the following: serve as the richest source of the most concentrated form of stored energy in plants, seeds and animals, serve as the chemical medium for the electron transport system of mitochondria and the photophosphorylation system of chloroplasts. Some lipids are actively involved in oxidative metabolism, phosphorylation, decarboxylation and methylation. Transport of materials such as cations across the lipid bilayer of cell membranes form the structural framework of cell membranes. Transport of materials such as cations across the lipid bilayer of cell membranes form the structural framework of cell membranes, serve as electrical and heat insulators and shock absorbers. Some steroid lipids serve as the raw materials for the synthesis of sex hormones, bile acids, and vitamin D, E, and K. Some lipids serve as enzyme activators. Some lipids serve as enzyme activators and metabolic regulators. Protect internal organs from mechanical damage, non-shearing thermogenesis, and regulation of body temperature in endotherms. Brown fat deposition. Promote the intestinal absorption of fat-soluble vitamins A, D, E, K, protection of some internal organs by forming defensive pad around them. Lipid profile. Lipids are essential constituents of blood plasma and also the structural framework of all cell membranes. In blood plasma, they exist mostly as lipoproteins. The concentration of the different kinds of lipids in blood plasma is called lipid profile. The concentration of the different kinds of lipids in blood plasma is called the lipid profile. Lipoproteins are tiny micellar spherical particles visible only through an electron microscope. The lipoproteins of human blood can be separated by ultracentrifugation. Based on their sedimentation coefficient in ultracentrifugation, they can be classified into several groups such as high density lipoprotein, low density lipoprotein, and intermediate density lipoprotein, very low density lipoprotein, etc. Chylomicrons are also there. In addition to this, cholesterol, phospholipid, etc. also form part of the lipid profile. Chylomicrons are protein-coated particles of triglycerides and very small quantity of cholesterol. The density of proteins and phospholipids depends on the quality of the lipids present in them. VLDL has only very little of lipids and HDL has high percentage of lipids. The clinical estimation of the various lipoprotein constituents of blood plasma is usually done after night fasting. Estimation of the lipid content of blood plasma is very important in the diagnosis of several diseases because high lipid level in blood are the major reasons for several cardiovascular problems. For example, high blood cholesterol level, hypercholesterolemia, is a major contributing factor for atherosclerosis, hardening, thickening, and narrowing of blood vessels, arterial blocks, ischemic heart diseases, stroke, etc. The normal lipid profile is phospholipids 150 to 200 mg, triglycerides 75 to 150 mg, cholesterol 50 to 200, HDL high density lipoprotein about in males 30 to 60 mg, in females 35 to 75 mg, LDL low density lipoprotein age 15 to 13 50 to 150 mg, age 30 to 40 75 to 180 mg, age 40 to 50 90 to 200 mg. So lipids, let's have a quick summary of lipids. Lipids are heterogeneous group of organic compounds insoluble in water and soluble in nonpolar organic solvents such as ether, chloroform, and benzene. Lipids are hydrophobic in nature due to the predominance of hydrocarbon chains in their substructures. Lipids widely occur in plant and animal kingdom. Lipids are formed of carbon, hydrogen, oxygen, occasionally nitrogen and phosphorus, which forms the framework for the structure and formation of living cells. Lipids are generally esters of fatty acids and alcohols. Fatty acids are aliphatic hydrocarbon chains with a carboxyl acidic group at one end, CH3, CH2, and COH. They are amphipathic molecules since they contain both polar and nonpolar groups. They may be saturated or unsaturated. A fatty acid is saturated if there exist no C double bond C bonds. Unsaturated fatty acids do contain C double bond C bonds. A glycerol molecule is made up of 3 carbon atoms with a hydroxyl group attached to it and H atoms at the remaining portions. This is a straight chain trihydroxy alcohol CH2OH, CH2OH, CH2OH. It has 3 linkage sites for esterifying 3 fatty acid molecules. The linkage occurs between the acidic carboxyl group of fatty acid and alcohol hydroxyl group of glycerol with elimination of water molecule. The linkage is ester linkage. The products of ester linkages are glycerides. Glycerol and fatty acid are linked by ester bond. So when we classify lipids, Bloor has proposed the following classification of lipids. Like lipids are classified into simple, complex, derived, and miscellaneous. Simple lipids are fats and oils and waxes. Complex lipids are phospholipids, glycolipids, sulfolipids, lipoproteins, etc. ಇದಕ್ಕೆ phospholipids, glycolipids, sulfolipids, lipoproteins. So derived lipids are of two types, namely fatty acids and alcohols. Miscellaneous lipids are aliphatic hydrocarbons and terpenes. So when we learn about simple lipids are homolipids, esters of fatty acid with various alcohols. Fats, esters of fatty acids with glycerol. Oils are fats in the fluid state, so they are called triglycerides. And waxes, esters of fatty acids with higher alcohols other than glycerol. Then fats and oils, we will be learning about fats and oils in detail. They are esters of fatty acids with glycerol. They are enormously found in nature. Fats and solid and oils are liquid at Fats are solid and oils are liquid at 20 degrees Celsius. Fats and oils are triglycerides or triglycerol. It is a fully esterified glycerol. The chemical structure of fat triglyceride consists of 3 different molecules of fatty acids with 1 molecule of glycerol. So, triglycerol. Triacyl glycerols containing the same kind of fatty acids in all the 3 positions are called simple triacyl glycerols or homoglycerides, and those containing 2 to 3 kinds of fatty acids are mixed triacyl glycerol or heteroglycerides. Most natural fats are mixtures of simple and mixed. Most natural fats are mixtures of simple and mixed. In triacyl glycerols, free acidic and basic groups are absent, so fats are called neutral fats. Fat and oil are nonpolar and hydrophobic because OH group of glycerol and COOH of fatty acid are involved in ester linkage and not involved in H bonding. Triglyceride is formed between 1 glycerol and 3 fatty acid molecules. Now, waxes. They are esters of fatty acids with higher alcohols and formed as secretions, which are mostly protective in function by many animals. Esters of long chain fatty acids and long chain non-glycerol aliphatic and monohydroxy alcohols. They are mixtures of esters and ethers derived from higher fatty acids and higher monobasic alcohols. They resemble the fats and usually solid. In human body, commonest waxes are esters of cholesterol. Waxes are highly hydrophobic and resistant to atmospheric oxidation. They form a water repellent protective coating. It protects plants from water loss and abrasive damages. Waxes are more solid and hydrophobic than fat and oil due to large hydrocarbon portion. R1OH monohydroxy alcohol is there. So beeswax, this is a mixture of esters, free fatty acids, alcohols and hydrocarbons. Its major constituent is myricyl palmitate, which is an ester of myricyl alcohol and palmitic acid. Next is the carnauba wax. This is a plant wax found in leaf cuticle of carnauba palm. This is a plant wax. Carnauba wax is a plant wax found in the leaf cuticle of carnauba palm. This is a sort of plant wax. Okay, it is also a complex mixture of esters, free fatty acids, alcohols and hydrocarbons. It is harder than beeswax and melt at high temperature. Sperm whale oil. This is an oil from the herd of sperm whales. Nearly 75% of it is wax. Now, lanolin or wool fat. This is a protective coat over wool fibers. It is a complex mixture of free and esterified cholesterol and sterols, lanosterol and agnosterol. It can take up water without dissolving. This makes it valuable as a medium in the preparation of ointments and cosmetics. So now let's learn about compound lipids. They are esters of fatty acids containing groups other than alcohol and fatty acids. They contain atoms other than CHO, most notably PS and N, that is phosphorus, sulfur, and nitrogen. Compound lipids or heterolipids are the lipids which remain linked with non-lipid molecules such as proteins, oligosaccharides, and phosphates. Phospholipids, phosphatides. Phospholipids are esters of fatty acids with alcohol containing an esterified phosphoric acid and a nitrogen base. They are present in large amounts in nerve tissue, brain, liver, kidney, pancreas, and heart. They are heterogeneous group of polar lipids widely distributed among bacteria, plants, and animals. They are amphipathic molecules with a polar hydrophilic head and a non-polar hydrophobic tail. The head is formed of negative phosphate group and positive alcohol group. Tail is formed of fatty acids. Glycerol forms the core constituent. Most of them are derivatives of phosphatidic acid. Two major groups, phosphatidylserines and sphingomyelins, are there. Now, phosphoglycerides, glycerophospholipid. Glycerophospholipids or phosphoglycerides are glycerol-based phospholipid. They are the main component of biological membranes, commonest phospholipids. They are fatty acid esters of glycerol phosphate. It is a diglyceride formed of two fatty acids, a glycerol and phosphate group complex. There are several kinds of phosphoglycerides. Phosphatidic acid and phosphatidylglycerol. Phosphatidic acid are anionic phospholipid important to cell signaling and as an intermediate in the synthesis of triglycerol and phospholipids. Hydrolysis gives rise to one molecule of glycerol and phosphoric acid and two molecules of fatty acid. Constitutes 0.25 of phospholipid in bilayer. Phosphatidylglycerol is a glycerophospholipid found in pulmonary surfactant. Cardiolipin, which is found in mitochondria, is formed from phosphatidylglycerol. Lecithins. They are phosphatidylcholine is the another alternative name of lecithins. They are choline derivatives of phosphatidic acid. They contain glycerol, fatty acids, phosphoric acid, and choline, nitrogenous base. Contain acidic COOH group of phosphoric acid and basic OH group of choline. So, exist as sweeter ions. That is the peculiarity of lecithins. Lecithins generally contain a saturated fatty acid at alpha position and an unsaturated fatty acid at beta position. Role in structural organization of protoplasm and cell membranes. So, cephalins will be there, phosphatidylethylamine. Ethanolamine derivative of phosphatidic acid, structurally similar to lecithin. Choline replaced by ethanolamine, also forms sweeter ion. Major role in organization of protoplasm and cell membrane. Cephalins are particularly abundant in brain, occur in tissues in association with lecithins. Phosphatidylserine. A cephalin-like phospholipid found in tissues. Phosphatidylinositol found in brain, liver, heart, and soya bean contain no base but have inositol in its place. More acidic than other phospholipids. Plasmalogens. Plasmalogens, commonly called ether lipids, they possess an ether link in a position instead of ester link. The alkyl radical is an unsaturated alcohol. These are the contents of brain and muscle. Structurally, this resembles lecithins and cephalins, but give positive reaction when tested for aldehydes with Schiff reagent after pre-treatment of phospholipid with mercury chloride. Next is the sphingomyelins. They are derivatives of sphingosine. Sphingosine is a long chain unsaturated nitrogen containing amino alcohol. Contain a complex amino alcohol sphingosine only found in animal tissues, a fatty acid, a phosphoric acid, and choline. No glycerol is present, abundant in blood, plasma, brain, and nerve tissue. They form an essential constituent of myelin sheath of Modulated nerve fibers, sphingomyelins, at least three types are known. So, biological roles of lipids. Energy storage. The excess energy from the food we eat are digested and incorporated into adipose tissue or fat tissue. Most of the energy required by the human body is provided by carbohydrates and lipids. Glucose is stored in the body as glycogen, while glycogen provides a ready source of energy. Lipids primarily function as an energy reserve. Fat vacuoles can be seen and cytoplasmic rim of a fat cell is there. Nucleus of a fat cell can be seen and a capillary and a areolae is there. Fat cells are there. Glycogen is quite bulky with heavy water content, thus the body cannot store too much of it long of glycogen. Alternately, fats are packed together tightly without water and store far greater amount of energy in a reduced space. A fat gram is densely concentrated with energy. It contains more than double the amount of energy in a gram of carbohydrate. Unlike other body cells that can store fat in limited supplies, fat cells are specialized for fat storage and are able to expand almost indefinitely in size. So, lipids are part of the cell membrane structure which help maintain cell membrane fluidity and flexibility. In the case of regulation, we are going to learn the importance of lipids in the field of regulation. Lipids are part of the cell membrane structure which help maintain cell membrane fluidity and flexibility. Triacyl glycerols control the body's internal climate with the goal of maintaining constant temperature. Those who don't have enough fat in their bodies tend to feel cold sooner. Triacyl glycerols also help the body to protect and regulate hormones. Adipose tissue secretes the hormone leptin which regulates appetite. Triacylglycerols also help the body to protect and regulate hormones. Like adipose tissue secretes the hormone leptin which regulates appetite. In the reproductive system, fatty acids are required for proper reproductive health. Women who lack proper amounts may stop menstruating and become infertile. So leptin is there, fat cells, leptin, hypothalamus, hormones and cytokines, food intake. Adipose tissue secretes the hormone leptin which regulates appetite. Omega 3 and omega 6 are essential fatty acids helps to regulate cholesterol, blood clotting and control inflammation in the joints, tissues and bloodstream. Fat also play important functional role in sustaining nerve impulse transmission, memory storage and tissue structure. Omega 3 versus omega 6 rich foods, walnuts, hemp seeds, soybeans contain equal amount or they equally contain omega 3 and omega 6. Omega 3 is greatly present in salmon, mackerels, flax seeds, chia seeds, and oysters. And omega 6 is present in peanut butter, almonds, avocado oil, cashew, sunflower seeds. More specifically, in the brain, lipids are focal to brain activity in structure and function. They help to form nerve cell membranes, impulse and insulate neurons and facilitate the signaling of electrical impulses throughout the brain. Insulating and protecting. In the field of insulation and protection, within the body there are two types of fat, visceral and subcutaneous fat. Visceral fat surrounds vital organs such as heart, kidney and liver. Subcutaneous fat or fat underneath the skin insulates the body from extreme temperatures and help keep the intestinal climate under control. Subcutaneous fat pads of our hands prevents friction as these areas subsequently come in contact with hard surfaces. Visceral fat surrounds visceral organs such as heart, kidney and liver. Subcutaneous fat or fat underneath the skin insulates the body from extreme temperature and helps keep the internal climate under control. Subcutaneous fat pads of our hands prevents friction as these areas frequently come in contact with hard surfaces. It also gives the body the extra padding required when engaging in physically demanding activities such as ice or roller skating. horseback riding or snowboarding. Next is the transporting function field. Fat-soluble nutrients are important for good health and exhibit a variety of functions. Vitamins A, D, E, and K of the fat-soluble vitamins are mainly found in foods containing fat. Fat-soluble nutrients require fat for effective absorption. These vitamins are best absorbed when combined with foods containing fat. Fats also increase the bioavailability of compounds known as phytochemicals, which are plant constituents such as lycopene found in tomatoes and beta carotene found in carrots. Phytochemicals are believed to promote health and well-being. As a result, eating tomatoes with olive oil or salad dressing will facilitate lycopene absorption. Other essential nutrients such as essential fatty acids are constituents of the fats themselves and serve as building blocks of a building block of a cell. Signaling. Lipid is very important in case of signaling. Evidence has emerged showing that lipid signaling is a vital part of the cell signaling. Lipid signaling may occur via activation of G protein coupled receptors. Members of several different lipid categories have been identified as signaling molecules and cellular messengers. This include sphingosine 1 phosphate, that is a potent messenger molecule involved in regulating calcium mobilization, cell growth and apoptosis, diacylglycerol DAG, and the phosphatidylinositol phosphatase PIPS involved in calcium mediated activation of protein kinase C, the phosphatoglandin, the prostaglandins, which are one type of fatty acid derived eicosanoid involved in the inflammation and immunity, the steroid hormones such as estrogen, testosterone, and cortisol, which modulate a host of functions such as reproduction, metabolism, and blood pressure. Phosphatidylserine lipids are known to be involved in signaling for the phagocytosis of apoptotic cells or pieces of cells. Phosphatidylserine lipids accomplish phagocytosis by being exposed to the extracellular phase of the cell membrane after the inactivation of flipases. Flipases, rarely spelled flipases, are transmembrane lipid transporter proteins located in the membrane which belong to ABC transporter or P4 type ATPase families, which place them exclusively on the cytosolic side, and the activation of scamblasters which scramble the orientation of the phospholipids. After this occurs, other cells recognize the phosphatidylserines and phagocytosize the cells or cell fragments, exposing them. Phosphatidylinositol is a small lipid molecule composed of an inositol ring and two fatty acid chains connected through a glycerol backbone, a structure that allows PTDLNS to anchor on the cytoplasmic face of cellular membranes. PTDLNs are phosphorylated by a host of lipid kinases at the 3, 4, and 5 hydroxyl positions of the inositol ring, producing a variety of phosphatidylinositol and monophosphates, diphosphates, and triphosphate that are collectively known as phosphoinositides. The phosphorylations are removed by site-specific lipid phosphatases, enabling dynamic flux between lipid phosphorylation states. In general, phosphatidylinositol monophosphates are located and localized to intracellular membranes, endocytic vesicles, Golgi apparatus nucleus, while di- and triphosphates are found at the plasma membrane. Phosphoinositides are universal signaling entities that regulate cell activities through direct interaction with membrane proteins, ion channels, GPCRs, through membrane recruitment of cytosolic proteins containing domains that directly bind phosphoinositides such as plextrin homology, FIVE, WD40 repeats, FERM, PTB and PTZ domains, among others. By far the best studied phosphonosidyl is which is synthesized from Pi4 to P2 by Pi3K class 1 and dephosphorylated by P10. Both P13K class 1 and P10 are classic central mediators of receptor tyrosine kinase induced AKT signaling and are often mutated in many forms of cancer. In addition to its effect on cell proliferation, survival, and metabolism associated with AKT signaling, phosphoinositide signaling also induces cytoskeleton changes and actin remodeling and plays a role in clathrin-mediated endocytosis, vesicle trafficking, membrane dynamics, autophagy, cell division, cell migration, and UV stress response. Lipid, which acts as cofactors. Proteins can adjust their structure and function in response to shifting environment. Functional diversity is created not only by the sequence but by changes in tertiary structure. Lipid cofactors may enable otherwise unstable protein folding variants to maintain their conformation to form novel biologically active complexes. Unsaturated C18 fatty acid synthesis conformation are the cofactors that bind apo-alpha lactalbumin and form hamlet human alpha lactalbumin made less lethal to tumor cells. The complexes were formed on an ion exchange column, were stable in a molten globule-like conformation and had attained the novel biological activity. The protein fatty acid interaction was specific as saturated F18 fatty acids or unsaturated C18-1 transforms were unable to form complexes with apo-alpha lactalbumin as were fatty acids with shorter or longer carbon chains. Lipids are essential tissue-specific cofactors. Lipid act as a component of biological membranes. Eukaryotic cells feature the compartmentalized membrane-bound organelles that carry out different biological functions. The glycerophospholipids are the main structural component of biological membranes, as the cellular plasma membrane and the intracellular membranes of organelles in animal cells, component of biological membranes. The plasma membrane physically separates the intracellular components from the extracellular environment. The glycerophospholipids are amphipathic molecules containing both hydrophobic and hydrophilic regions that contain a glycerol core linked to two fatty acid derived tails by ester linkages and to one head group by a phosphate ester linkage. While glycerophospholipids are the major component of biological membranes, other non-glyceride lipid components such as sphingomyelin and sterols, mainly cholesterol in animal cell membranes, are also found in biological membranes. Important component of membranes of chloroplasts and related organelles and the most abundant lipids in photosynthetic tissues, including those of higher plants, algae, and certain bacteria. Plant thylakoid membranes have the largest lipid component of a non-bilayer forming monogalactosyl diglyceride and little phospholipids. How cholesterol is formed. Much of the cholesterol is located in cell membranes. It is also found in the blood in free form as plasma lipoproteins. Lipoproteins are complex aggregates of lipids and proteins that make travel of lipids in a watery or aqueous solution possible and enable their transport throughout the body. The main groups are classified as chylomicrons, very low density lipoproteins, and high density lipoproteins based on their relative densities. Cholesterol maintains the fluidity of membranes by interacting with their complex lipid components, specifically the phospholipids such as phosphatidylcholine and sphingomyelin. Cholesterol also is the precursor of bile acids, vitamin D, and sterol hormones. Prostaglandin formation. The essential fatty acids, linoleic acid and linoleic acids, are precursors of many different types of eicosanoids, including the hydroxyeicosanoids, prostanoids, prostaglandins, thrombox and prostacyclins, leukotrienes, and lipoxins, along with resolvins. This play an important role in pain, fever, inflammation, and blood clotting.